Let's now take a look at carbohydrate digestion, and this is going to involve the enzymes that are being secreted by the pancreas due to the dietary stimulation. Now on this molecule I have drawn here, notice that this is a large polymer of monosaccharides linked together by oxygens, which is an O-glycosidic linkage. This particular structure is starch, which is arguably our greatest energy source. Now, in order for us to be able to use this as an energy source, we have to digest this polysaccharide back down into its monomeric structure. And in order to do that, we are going to need the enzyme amylase. As we saw before, amylase is also secreted by the salivary gland, but the contributions to starch digestion by the salivary gland amylase is minimal compared to the pancreatic amylase because of quantity of enzyme. Now the amylase is going to be specific for this O-glycosidic linkage. Now notice that this is the number 1 carbon here. The adjacent monomer of glucose is the number 4 carbon, and the glucose monomers are linked together by an O-glycosidic linkage, and the O, the oxygen here in this case, is in the down configuration. So this is an alpha 1,4 O-glycosidic linkage. Amylase is specific for the alpha 1,4 linkage. Now recall the polymer cellulose. We looked at cellulose before. Cellulose has the same polymer structure of glucose, however the O-glycosidic linkage is in an up configuration, which makes it a beta 1,4. Amylase has no effect on cellulose because the amylase enzyme is specific for the alpha 1,4 linkage. What does digest cellulose? Well, that would be cellulase. But humans do not have the enzyme cellulase. And so therefore cellulose, even though it's a large, dense polymer of glucose, is useless for us as a fuel source. Now what can we use cellulose for? Well, cellulose is important in adding bulk to stools. So the presence of cellulose helps to add bulk and structure to the stool, to the feces. It also adds hydration, very important. Hydration because of all of the hydroxyl groups and oxygens in the ring structure and in the glycosidic linkages. These, recall, can form hydrogen bonds with water. And in doing so, the cellulose will bind a lot of water to it. So if you have bulk structure to the stool by having cellulose, that will hold water, and then that will prevent constipation. Now the other carbohydrates that we have in our diet, the next one we have to take a look at is called sucrose. And sucrose is a disaccharide and sucrose consists of fructose in a O-glycosidic linkage with glucose. So this is a disaccharide, so only 2 monomers hooked together. The enzyme that is specific to hydrolyze this is sucrase. Now sucrase, unlike amylase, amylase is made by the pancreas and secreted into the duodenum. Sucrase is an enzyme on the epithelial brush border of the intestine. So sucrase is a membrane-bound enzyme. And it's actually a double-headed enzyme. So if this is the epithelial cell plasma membrane, also called the enterocyte, we have an enzyme that crosses the membrane. It's a transmembrane protein. We have one domain. We have a second domain. This domain accounts for the sucrase activity that will digest the fructose-glucose disaccharide. This domain will have another enzyme that is called isomaltase. So we have two independent activities in one polypeptide that is bound to the plasma membrane of the intestinal border. Now, sucrase is obviously specific for sucrose. What is isomaltase? Well, let's go back to our starch molecule. We have our polymer of glucose linked together with our alpha 1,4 linkages, but notice this linkage here. This is a linkage between the number 1 carbon of this glucose monomer and the number 6 carbon of this glucose monomer. So therefore this, notice the down configuration of the hydroxyl, this is an alpha 1,6 O-glycosidic linkage. Of course, amylase is specific for alpha 1,4, so therefore amylase has no effect on this bond. Isomaltase, however, does. So the function of isomaltase is to digest, hydrolyze, this alpha 1,6 linkage, to release the upper portion, which is, again, another long polymer of glucose. And the alpha 1,6, once that's digested, then we have availability of all the rest of the alpha 1,4 for amylase to work correctly. The last carbohydrate we have to consider is the one that's found in many dairy products, and this is lactose. And lactose is also a disaccharide. And this disaccharide is two sugars of galactose and glucose. The enzyme that is specific for this linkage, of course, is lactase. Lactase is similar in properties, physical properties, as the sucrase isomaltase enzyme, in that it is also found on the plasma membrane of enterocytes. So the lactase enzyme is a separate structure, but it's also membrane bound by a transmembrane sequence. Now, ending with lactose, we have to consider this is oftentimes a very difficult sugar because of lactose intolerance. And in the lecture notes, I provide you with a little bit of the frequency of individuals in different ethnicities that have lactose intolerance. And what this is the inactivation of lactase, the gene for lactase. In many situations, this gene is active in childhood, but then for some reason that's still unknown, the gene gets turned off in adulthood. Therefore, the individuals have no lactase and they are not able to digest lactose. So if we are not able to digest lactose, there is another organism in our gut that allows us to digest this, and these are bacteria. The bacteria will digest the lactose into its galactose and glucose monomers and then ferment. down to lactate. They will use this as an energy source for growth. Now, part of this is gas and bloating. And because we have lactose in our intestine, which is able to bind water, we have lactate, which is also hygroscopic, able to bind water. We have an increase in water retention in the colon. Because of this water binding properties, we increase our chances of having watery diarrhea. The other problem here is that by increasing the amount of water, increasing gas production because of the bacteria, these two effects will increase colonic motility. And these are the end results of lactose intolerance. Now, of course, there are ways to resolve this. We do have lactose-free dairy products that are now available, or enzyme supplementation during dietary consumption of actually ingesting the lactase enzyme in a pill form.